We have a fabulous lineup for you. On the panel, we have Professors Mary Herbert and Doug Turnbull, who both pioneered the use of mitochondrial donation. We have Liz Curtis, who founded the Lilly Foundation. And we have Professor Catherine Mills, a bioethicist. So it gives me great pleasure now to introduce tonight's first speaker, Professor Mary Herbert. Mary is a pioneer of mitochondrial donation and lead author of the first study reporting the embryological and reproductive aspects of the use of mitochondrial donation treatment in the UK. Mary is Professor of Reproductive Biology at Monash University's Biomedicine Discovery Institute and Clinical Research Lead at the mitoHOPE Pilot Programme for Mitochondrial Donation in Australia. She's a contributor to books including Reproduction, Clinical Reproductive Science and Biology, and The Pathology of the Oocyte.
Those of you who are not so familiar with this field, I'm going to give a quick background before I start talking about our results. So the key players in this is the fertilized egg, which you see here at the center, on the center of the screen. And in the center of this egg, you see two pronuclei, and that each contains a nuclear genome, half of which comes from the egg and half of which comes from the sperm. And this egg, fertilized egg, also contains mitochondrial DNA, which is inherited only from the egg. And it contains about 200 to 500,000 copies of mitochondrial DNA. And I'll talk more about the mitochondria in the next slide. But just to say that over the course of 5 to 6 days after fertilization, this fertilized egg develops to form undergo cell division, and a proportion of these embryos will go on to form a blastocyst. And a blastocyst is an important developmental milestone because that's the stage at which an embryo can implant in the uterus. And here in the inner cell mass, which you'll see at about the 2 o'clock position, it contains a small number of cells that will then go on to form the fetus. So genes encoded by the mitochondrial DNA are essential for energy production, and this energy is in the form of ATP. Mitochondrial DNA is contained in the matrix of the mitochondria, and it encodes just 37 genes. 13 of them are subunits. 13 encode subunits of the respiratory chain complex, which is the machine that generates the ATP. And if a woman carries harmful mutations in her mitochondrial DNA, this can cause deficits in energy production, and she is at risk of transmitting disease to her children. But inheritance of mitochondrial DNA disease is really characterized by unpredictability, which makes for profoundly difficult reproductive decisions. So here we go. This is a human egg here, stained for, we've used a fluorescent label for the mitochondria here in red, and we've stained the DNA in green. And you can see the nuclear DNA here in the 3 o'clock position, and then all through the egg, we see the mitochondrial DNA. And for a woman who carries mutations, the mutations can be present in all copies of the mitochondrial DNA or in some. So in the case of being present, in the case of coexistence of unaffected and mutated mitochondrial DNA, this is known as heteroplasmy. And the level of heteroplasmy can vary widely between eggs from the same woman. If the mutation is present in all copies of mitochondrial DNA, this is known as homoplasmy. And so the mutation is transmitted in full to any children of that woman. In this case, it's also difficult to predict the risk of disease because even though all of the mitochondrial DNA carries the mutation, the disease penetrance is variable. And you'll hear more about that in Doug's talk. So as I said, the risk of disease in children born and the unpredictability of it makes for really difficult reproductive decisions. So we developed a program in collaboration with the mitochondrial team in Newcastle of assisted reproductive technologies to reduce the risk of transmission. And we offer pre-implantation genetic testing. And as you all probably know, pre-implantation genetic testing has been around for a long time to detect nuclear genetic defects, but it's used in a number of centers also for detecting mitochondrial mutations. And women will undergo ovarian stimulation and produce a cohort of embryos, which can then be tested. We can remove cells from the embryo and sent to the mitochondrial diagnostic lab for testing, and it enables us to identify embryos with low mutation loads. And in this way, we can select the embryos with low mutation load and thereby reducing the risk of disease in cases of heteroplasmy. But of course, PGT is not useful in cases of homoplasmy or indeed in some cases of high heteroplasmy. So in these cases, the technique of mitochondrial donation, also known as mitochondrial replacement, was developed. And this involves transplanting the nuclear genome from the egg of an affected woman to an enucleated egg from a non-affected donor. And it's suitable for women who are unlikely to benefit from PGT. So mitochondrial donation, this nuclear genome transplantation, can be performed before fertilization. And in that case, you use special optics to see the spindle, which you can see in the schematics here with the chromosomes. And this is called maternal spindle transfer. If it's performed after fertilization, this is called pronuclear transfer, which involves removing the pronuclei, which I spoke about earlier. Now, in Newcastle, we have focused on the pronuclear transfer technique because that was the one that was most widely used in animal models when we started doing this work. And I'm just going to show you a movie of my colleague here, Louise Hislop, doing this. And these techniques were optimized over the course of a very long time. We published proof of concept work in 2010 and preclinical work in 2016. And you see Louise here is removing one pronuclease at a time. And she briefly exposes them to a fusogen, and then she places them under this outer shell, the zona of the enucleated donor egg. And then they fuse quite quickly over the next hour. And then this embryo goes on to develop. So now to move to the sort of clinical interventions that are involved in these treatments. So this scheme is showing you the normal ART-related procedures that a woman would undergo. We have ovarian stimulation, which involves hormone injections to stimulate growth of multiple follicles. Then the eggs are retrieved after 11 to 14 days of injection, and then they're fertilized in the lab, usually by ICSI, which involves injecting sperm into the cytoplasm, usually by ICSI for our purposes here. And then the embryos are cultured for 5 to 6 days, and those that are of sufficiently good quality will be used for fresh embryo transfer, or they will be frozen by vitrification for future use. So the additional steps in PGT involves removal of a cell to test the mitochondrial DNA mutation load. And we do this on day 3, or in this series, we did it on day 3 of development when embryos contain between 6 and 8 cells. And we focused on this elastomere biopsy because that's what had been used in all of the PGT publications for mitochondrial DNA disease. The additional steps for pronuclear transfer, patients' eggs are vitrified, and that scheme of things is based on the preclinical research. And then we have the donor egg collection, and then we have PNT performed between vitrified patient eggs and fresh donor eggs. So to show you the number of patients who have come through for treatment, we have 39 women underwent ovarian stimulation for PGT treatment. They all had egg retrieval and ICSI. 37 of them had embryos that were suitable for biopsy, and of those, 31 had embryos replaced. And the drop-off here is largely due to having no embryos with sufficiently low mutation load for replacement. And then in 16 of those women, we had a clinical pregnancy. Looking at pronuclear transfer treatment, we require case-specific approval for each of these cases. And so far, we've obtained approval for 32 women, and 25 of whom have undergone ovarian stimulation, and 25 had eggs collected and vitrified. 22 of those have so far undergone at least one ICSI procedure in preparation for pronuclear transfer, and 19 have had at least one pronuclear transfer treatment. The drop-off here is because of abnormal fertilization. And embryos were available for transfer or for freezing in 18 women, and 8 of them became pregnant. had a clinical pregnancy. So I'm just going to talk you through briefly the sort of pertinent embryology findings. What we found was that the pronuclear transfer eggs had reduced fertilization compared with donor eggs. So it was 48.5% compared with about 64%. And these donor eggs were fertilized with eggs from the same source, with sperm from the same source. So this tells us that the reduced fertilization here was a problem with the eggs. And the pronuclear transfer fertilization was also reduced compared with the PGT patients. Now we don't know what, so these pronuclear transfer patient eggs were vitrified, and that has a small effect on fertilization. But the numbers are still small, but there seems to be a variant-specific effect on fertilization as well. So depending on the type of mutation that's present in the eggs, there seems to be an effect of some of these on fertilization outcomes, but the numbers are really too small yet. So because of this reduced fertilization, 3 pronuclear transfer patients had no normally fertilized eggs available for pronuclear transfer. And looking at the survival of these eggs, so we had PNT was performed in 160 eggs from 19 patients. And the points of loss here usually is we get lysis of the karyoplast during the PNT procedure. This occurred in about 20% of eggs. And then overnight, we had a further 4% degenerating. So in all of the 160 eggs, we had 122 surviving. And this graph here is on the right is showing you the number of intact eggs, pronuclear transfer eggs per patient, varying from 0 to 7, and 25% of patients had fewer than 2 intact eggs. So looking at embryo development, these pronuclear transfer eggs were cultured for 5 and 6 days, and the embryo development was equivalent between the pronuclear transfer and PGT groups. Development to the blastocyst stage. And then when we compare the quality of those blastocysts, we got a slightly increased proportion of top and good quality blastocysts after pronuclear transfer compared with PGT. So the embryo transfer outcomes of the PNT and PGT patients, about 80% of them had embryos available for transfer. And the outcome of those transfers, this is per transfer here, we had 40 transfer procedures of pronuclear transfer embryos and 51 for PGT. And the positive pregnancy test was about equivalent between the 2 treatments. But by the time it got to clinical pregnancy, which is defined as the presence of a fetal heartbeat at 6 weeks, it was significantly reduced in the pronuclear transfer group. So there was a drop off. There's an increase in what we call the biochemical pregnancy rate. That's loss of embryo loss after a positive pregnancy test. But when we look at the pregnancy rate, clinical pregnancy rate per patient coming through the door, it was about equivalent between the 2 treatments, the 36% for pronuclear transfer and 41% for PGT. And 3 of the pronuclear transfer cases, or 3 of the PGT cases had miscarriage, whereas none of the pronuclear transfer patients had. So in summary, we had 8 live births and 1 ongoing pregnancy after PNT, 4 males, 4 females, 18 live births after PGT, 9 females, 9 males, and no congenital abnormalities reported at birth. So looking at did we reduce the level of mitochondrial DNA mutation? Here we have the 7 women who became pregnant. There's 1 set of twins in this PNT group. And these are the mutations that we treated. This is the level in the second column here is the maternal level. And this pre-treatment enucleated egg is the level that would have been in the patient egg had we not performed pronuclear transfer. So the patient egg, in 6 cases, in 5 cases, the egg was homoplasmic, and in 2 cases, it was heteroplasmic. And then when we look at the levels in the neonatal blood, it ranges from undetectable to 16%. So in 5 of the 8 babies, we had undetectable levels. So the reduction in mutation load overall was between 77 and 100%. In 6 newborns, we had 95 to 100% reduction, and in 2, we had 77 to 88%. And just to summarize, we had 37 women have undergone embryo biopsy for PGT, involving 12 different heteroplasmic variants, ranging from undetectable to 98% in maternal levels. 18 babies were born, and the heteroplasmy for maternal pathogenic variants, which based on detection in 10 infants, was undetectable to 7%. 19 women underwent at least one round of pronate transfer for 8 different variants, 4 homoplasmic, 3 heteroplasmic. And the heteroplasmy here ranged from 79 to 89%. 8 babies born, 1 ongoing pregnancy, heteroplasmy ranging from undetectable to 16%. And just the ongoing work in PGT is to test the reliability of trophectoderm cell biopsy compared with day 3 biopsy. And then for PNT, we're working towards increasing the yield of viable embryos and reducing mitochondrial DNA carryover. So with that, I'd like to thank you for your attention, and I have just named a small subset of the large team from the fertility team and the mitochondrial team and the research team that were involved in this work.
I'm just going to have one question because I shouldn't really know the answer to this, but I've never felt I do. I've always seen your slides or Doug's slides, and you see how you explain the procedure, and you see the cells as like the size of a ping pong ball. And so in my imagination, I suppose, they stay that. Obviously, I know they're much smaller, but can you give us an idea of how tiny, these cells are that you're working with?
So the egg is about the width of a human hair, right? And
Right.
then, so that's about 110, 120 microns usually. And then the pronuclei are in the range of usually 25 to 30 microns. So pretty tiny.
About, yeah. So less than the width of a split end, really. If you're talking about human hair at the beginning of the sort of way of imagining it. Yeah. Wow. Yeah. Yeah.
Yeah,
That really, then you
don't,
really have another an
don't breathe.
increased level. Sorry.
Yeah. Yeah.
So you have a sort of increased level of respect for the people that are handling those tiny things and manipulating them. It is quite incredible. Okay.
Yes, I was going to say generally they don't breathe when they're doing it, so
Yeah.
it's just concentration.
So our next speaker is Professor Sir Doug Turnbull. Doug is a pioneer of mitochondrial donation and lead author of the first study reporting maternal and child health outcomes following the use of mitochondrial donation treatment in the UK. He's an Emeritus Professor of Neurology at Newcastle University and patron of the Lilly Foundation. He's a contributor to books including Diagnosis and Management of Mitochondrial Disorders, The Mitochondrion in Aging and Disease, and Biology and Pathology of the Oocyte.
I'm really talking about where this all started, and this started with this remarkable lady who many of you who've been involved in this over the years will have heard about Sharon's story, and some of you will have indeed met Sharon. And this always brings us back to the point that the whole point of doing this was because we kept meeting patients, families in our clinic who'd lost children due to mitochondrial DNA disease. And Mary's explained the genetics around this. And this part of the talk is all going to be around the fact that we are looking at what we do in relation not to the embryology, but in terms of the patients. So Mary's already explained that mitochondrial DNA is present in multiple copies in all cells. And what I'm going to do now is talk about the clinical aspects of this. So as Mary explained that you can actually have either all normal mitochondria, a mixture of mutated and wild type mitochondria called heteroplasmy, or all mutated. And in relationship to disease, heteroplasmy, especially in patients who've got the mutations which we're going through in terms of mitochondrial donation, the level of heteroplasmy, the level of mutated mitochondrial DNA that you have to have before you get disease is really quite high. And this comes back to a point I'm going to come back to later on when we talk about the success or benefits of mitochondrial donation. So as Sarah said, this all really started many, many years ago, but the help of Sarah, all the people at PET, the Wellcome Lilly Foundation in particular, and very grateful to Liz, and so many people in the UK, getting this through Parliament was a major achievement back in 2015. But in some senses, that set the starting gun going, because getting it through Parliament was only part of it. Obviously, huge amount of work had been done by Mary and the team in relationship to developing the technique. But it wasn't going to be any good if it passed the law and it was through, and we had a technique, if we couldn't set up a clinical service. And it's just incredibly grateful that we're very fortunate we've got an NHS, and we're very fortunate we've got a lot of very, very good people in NHS. And I know NHS England is currently being disbanded, but there was, in the highly specialised services, there was a real will to see this happen. So a combination of funding through the NHS and some support via Wellcome really meant that we could move forward with a clinical service. And of course, this was something entirely new. There wasn't anything quite like this. It was under intense scrutiny, far more, I think, than any other IVF procedure. So we had to set and train a clinical service. And one of the things that you should do when you set up a clinical service is ask patients. And again, we did this very much in partnership with the Lilly Foundation, that we really want to develop a service that would actually be fully appropriate for the patients that we're dealing with. And one of the ways you can do this is by holding focus groups. Multiple focus groups were held around all sorts of different aspects of providing the service. And this was one which was particularly about the follow-up of children born. And, it was really instructive about, and really was one of the things that really changed the way in which we actually decided to run the clinical service, was what the patients wanted. So they wanted access to specialist care. So if there's a problem with a baby, who do you turn to? Well, having access to that specialist care was important. About confidentiality. The world's changed since Louise Brown, and the last thing that patients want to do is be bothered by various aspects of either press or social media, and they didn't want their children to be treated like an experiment. So again, you're getting a way in which we might develop the service. So this was what was put into practice back in 2017. There's very much a referral. Obviously, you've got to be referred into the clinic, and then 2 specialist clinics before patients then go forward with the 2 techniques that Mary's discussed, PGT and mitochondrial donation. And it's interesting just looking at some of the numbers. We've just had, by the time of publication, just under 200 people have been referred. About 160 went to the reproductive advice clinic, 133 to the mitochondrial assisted reproductive technology clinic, both of which I'll explain a little bit more about. But as Mary said, 39 went to PGT and 32 to MT. The reason here is that there's quite a bit of people that, out of the 133 seen at the reproductive technology clinic, many of those women were going there to get advice and to get ideas about, and they weren't necessarily wanting to proceed with with this pathway at the present moment, but will be part of the future. And one of the things that we've encouraged throughout is that the people that are seen in our clinics are actually encouraged to get advice earlier rather than later. So this is a reproductive advice clinic, and really this is a triage for referrals. It does a number of things. It assesses the fitness for fertility, but also discusses the genetic risks and the various complications of going forward with any sort of IVF procedure. Very important, because don't forget, the women themselves are patients who have been seen in our clinic, and some of them have certain significant disease. And obviously we're providing that information, and then patients will then proceed to the mitochondrial assisted reproductive technologies clinic, or the MARC clinic as we call it. And what we've done is, because most patients come from different parts of the country, the patients will be seen in the morning in the initial clinic, and then in this, then there'll be what we call an MDT, which is a group meeting about the patients, and then they're seen in the reproductive technologies clinic. And there they're seen by an IVF clinician and a mitochondrial clinician together, so that they can have a more in-depth discussion if they're wanting to proceed with one of the IVF-based techniques. And they will discuss at that time mitochondrial donation, PGT, and egg donation. And then they will be provided with that information and giving an opportunity to go away and think about it, and then come back to the clinic. So throughout this process, there's a lot of counselling going ahead. And I think it's really important that one of the real safety checks that's put into place is, if a patient wants to proceed with mitochondrial donation, this is done on an individual basis, that an application has got to go through to the HFEA, and then that is assessed by a specific clinic at the HFEA, having an external advice from mitochondrial experts. There's a senior clinician, geneticist, leads that clinic, and they have an advisor to the committee who's also a mitochondrial expert. So only patients who we think are appropriate and then are approved by the HFEA can go forward for mitochondrial donation. So what does the pathway look in practice once they've decide to go forward? Mary and Louise have worked their magic. What then is the way forward? And this is what actually happens, and this is the pathway that is done through pregnancy. Clearly, a lot of the things that are done during pregnancy in the UK are absolutely standard and very appropriate for women who are undergoing mitochondrial donation. Women will have these 3 ultrasound scans. The things that are different are that for patients who have undergone mitochondrial donation, we strongly advise an obstetrician-led antenatal care, so that they are more closely monitored. And as you can see by the green triangles, throughout the pregnancy, they're contacted by people from the clinical team to see how patients are doing as regards their pregnancy. So it's closely monitored, but we hope not too intrusive. So there have been 7 pregnancies, 8 babies, as Mary said, 1 twin, and 6 have had no complications. There was one pregnancy where the lady, due to a mitochondrial disease, had a pre-existing cardiomyopathy, and during preparation for embryo transfer, developed paroxysmal atrial fibrillation, an abnormal beating of the heart, for which she required specific treatment. This lady also developed a severe and serious complication of pregnancy called extreme hypertriglyceridemia, or very high blood fats. And to just give you a concept that the level of triglyceride or fat in her blood was 77 with a normal range of up to 2. I mean, her blood looked like pink milk, in effect. It was a extremely concerning time for everybody. It's an extremely rare complication of pregnancy with a high morbidity rate, who is superbly looked after by the clinicians who not based in Newcastle. And she was treated with a low-fat diet and responded extremely well. I want to stress that I have never, having looked after hundreds of patients with mitochondrial disease, I've never come across this before as a complication of pregnancy, and it's never been described, as far as I'm aware, associated with IVF. So this is just a very unfortunate incident. And this, of course, as all things happen, this was actually the first pregnancy. So you can see how our anxiety levels were high after this, never been seen since, and it's just, we suspect, as you'll find out later, there must be another genetic cause for this. So what about the pathway of follow-up for the children? Well, again, what the families wanted was that this was appropriate, not too invasive, but carefully monitored. And again, those of you that recently had children in the UK, or in my particular case, having had grandchildren in the not too distant past, you'll know that they go through an extensive check with the GP, with vaccinations, with health visitors. And at each time we record those findings. But throughout the period, certainly up to 18 months, the patients, the parents, or the clinicians can contact the mitochondrial team. My colleague Bobby McFarland is a professor of pediatric neurology and a world expert in pediatric mitochondrial disease, and an excellent clinician, and they can always get Bobby's advice about the way forward. We have brought in a 3 to 6 month cardiology review, and at 18 months, they undergo an intensive assessment, which I'll explain. We check for whether or not the level is of whether or not they've got a change in their mitochondrial load at 18 months. And we're introducing a 5-year review. The reason for assessing at 18 months is that most children are walking and have specific neurodevelopment at that stage, and 5 years is just before people go to school in the UK, and again another good time to check whether or not the development's normal. So only, well, 1 at the time, only 2 now have gone through to the 18-month stage. So a lot of these babies are still quite young, something that obviously we're closely monitoring, and the children will go through the various stages. This slightly busy slide is just to show you some of the detail that Mary's already described, but you can see that babies on the whole went pretty close to term. The twins were born somewhat early in terms of weeks. The mode of delivery was either cesarean section or normal vaginal delivery. Birth weights were within normal limits, and the APGAR scores, which are the scores which are taken immediately after birth, were fine, with good APGAR scores at 10 minutes. So, as Mary said, there were no congenital defects noted, and certainly the births were thought to be completely normal. And Mary's already described the level of variant that's been found. I just want to stress again, I mentioned about the level of heteroplasmy in clinical disease. So these are again the variants that we've seen, the variants that have all the mutations which have patients who undergone mitochondrial donation, and you can see that there are these particular variants are the ones that have been looked at. 4 of them have got the 11, 7, 7, and 8 variant. So even though one of the children born has got a level of this mutant variant, actually we've only ever seen clinical disease with very, very high levels of the variant or homoplasmy. So even if you had 20%, the actual risk of getting clinical disease is negligible. And it's likewise with all the other variants that really you're only going to see clinical disease at very high levels. It is interesting that the one patient who's reached 18 months where we've checked the level in blood and urine at 18 months, that the level, if anything's gone down. So, which is, of course, reassuring. So I mentioned that the children undergo an intense assessment at 18 months. This is a structured developmental assessment, which is carried out by 2 pediatric clinical neurologists, along with hearing and vision tests. One child had undergone this at the time of the paper was published, another has now, and development seems to be completely normal. And what about the health problems? Well, 5 children have had no health problems at all. If you have young children, or you've known about, or if you've had young children or grandparents or whatever it is, children get illnesses. 1 child presented with fever, rigors, and a urinary tract infection, which resolved perfectly well with antibiotics. 1 had some self-resolving muscle jerks. They're called myoclonic jerks. Again, quite an unusual issue, clinical problem, but absolutely benign and has settled down and completely unrelated, I think, completely unrelated to the condition. 1 child had a high blood fat content, and that's the child that's mother had the extreme hyperlipidemia and has a disturbance of heart rhythm. Both have responded well to treatment. We don't think any of those complications, any of these symptoms are related at all to the mitochondrial DNA. And I think the most important thing is that these children are all well and meeting their developmental milestones. Now, one of the questions is, where do we go from here? Well, clearly, a very important part of this is that we continue this program and we keep a very close eye on the development of these children. Everything is looking very positive at the moment, and clearly one of the things that we want to do, and one of the things that this has done so far, is it's provided a great reassurance to the families that are potentially considering mitochondrial donation, because of course until this time there haven't been follow-up of any children that have been born previously due to mitochondrial donation. So at that point, I'd like to conclude. Like Mary, I'd like to thank the huge team of people that have helped in Newcastle, to both the IVF, both the fertility team and the mitochondrial team, but also more broadly, the support of the whole community, the patient community, organizations like PET and Welcome. This would not have been possible without the support of so many people. And it's really pleasing to be able to present such positive results from the first 8 babies born.
Liz is she's somewhat of a legend, really. She is the chief executive of the Lilly Foundation, which helps patients and families affected by mitochondrial disease and seeks to improve diagnosis via the Lilly Precision Medicine Diagnostic Project. Liz established the Lilly Foundation after losing her daughter Lily to mitochondrial disease. Liz was an integral part of the campaign for the UK law to be changed to permit the use of mitochondrial donation in treatment. And Liz first spoke for us at our very first PET event on the topic of mitochondrial donation back in September 2022, when we were all still full of Olympic spirit, I think.
I set up the Lilly Foundation after we lost our daughter Lily in 2007. She was just 8 months old, and she died of a mitochondrial disease. At the time when she was diagnosed, we'd never heard of mitochondrial disease, and we were completely in the dark, and there wasn't that much information available for patients and parents like us. It seemed that everything that we found was written for doctors and by doctors. And there were small pockets of research going on, and it felt like no real hope of treatments or cures back in that time. So as a family, we started something, and this has grown into a nationwide charity which now supports over 1,300 families living with mitochondrial disease in the UK. And everything we do is built around 3 key areas. To funding life-changing research, supporting families, and raising awareness. With our ultimate goal to find treatments for mitochondrial disease and hopefully one day a cure. When Lily was diagnosed back in 2007, we were told that it was probably unlikely in our lifetime we would ever get a genetic diagnosis for her. And I remember thinking, how on earth are we ever going to find a treatment or a cure for this condition if we can't diagnose it? So our first, sorry, this is going far too fast for me. I can't keep up. Our first diagnostic project, was to fund a gene testing program. And this was a groundbreaking project which helped diagnose over 70% of the families that were involved in this genetic testing. And this now forms part of the NHS standard genetic testing pipeline. And we now invested a further £1 million into advanced diagnostics to help those who haven't yet received a diagnosis by the NHS. As a charity, we've also funded over 20 research projects and we've helped to build big national partnerships, including the current 5-year initiative with the LifeArc and Muscular Dystrophy as partners in the LifeArc Centre for Rare Mitochondrial Diseases. However, I think it's fair to say that one of our proudest moments to date has absolutely been our involvement in the mitochondrial donation debate. As you say, Sarah, we first got involved when you invited us to talk in the event. It was 2012, not 2022, when our job was to come along and to talk about the real life impacts of living with mitochondrial disease. And I think I'm right in saying that whilst there was a lot of debates around the science around mitochondrial donation at the time, there hadn't been much input from the patient voice. And I think on that night in September 2012, myself and my colleagues spoke about how mitochondrial disease had affected us personally. We'd both lost our children to the condition. It turned our worlds upside down. And for the first time, people were really hearing how devastating the effects of mitochondrial diseases are on patients and on families. And the impact that night of the patient voice was evident. And it was following this that we were asked to join a working group alongside yourselves at PET, the Wellcome Trust, the Genetic Alliance, and the Newcastle teams. And our role was to ensure that those who had the power to vote on this bill had a true understanding of what it was like to live with mitochondrial disease and the devastating effects it had. So using the power of social media and obviously our contact database, we urged our community to contact their MPs to enlighten them of the realities of living with mito. We did our very best to reach as many MPs as possible and peers as possible, hundreds of meetings. I felt like I lived in Westminster at one stage, with MPs and peers, and alongside the working group, we hosted and took part in briefings, in drop-ins, with the purpose to educate on the impact of mito, but also to ensure that the MPs and the peers were educated on the rigorous process that had been put in place, to ensure that this technique had been well considered and was safe. And whilst we knew at the time that this technique wouldn't be beneficial to absolutely everybody who suffered from mitochondrial disease, because not everybody is eligible for mitochondrial donation. However, the support that we received from the community was overwhelming. Despite this, it didn't matter that the technique might not benefit everybody. What matters was that it was going to benefit some. And this was enough to get people moving. It was enough to get them engaging with their MPs and telling their story. And boy, did it make a difference. And I'm not taking anything away from the incredible science that this technique has been built on, but it became really clear in Parliament on the day of the vote that the patient stories and the interaction with their MPs was crucial to the way in which the vote went. Oh yeah, there we go. There's some quotes there that were taken from the Hansard at the date after the vote in Parliament. That we won't have time to read now, because otherwise I'll go over my 10 minutes, but they're there and I can forward them if anybody wants to see them. But also I felt like I couldn't do this talk without sharing this short video.
The eyes to the right, 382. The nose to the left, 128.
Now I think, that really shows that what it meant to those of us that were there in Parliament that day. We actually did get told off for cheering in the chamber, but I don't think we really cared. It really did mean so much to everybody who had put their time and effort into to be involved. And then there's a quote here from a patient too, who after we shared the recent news, he wrote to me with this quote. But once again, so to be able to share the recent news that mitochondrial donation had actually worked, and that there were 8 babies who have been born free of this disease, is without doubt the most positive news that we've ever been able to share with our mitochondrial community. For the first time, it offered real hope to those who are affected by mito, and it shone a big light on the disease as a whole. And these messages that we have received have just been incredible. And we look forward to a future of preventing mitochondrial disease for so many families. And Doug, you mentioned, the reassurance that this news has given. I've seen a huge impact already on families and young women who are looking to this technique to have babies in the future, and to know that it works, to have that reassurance is just absolutely incredible. So whilst we look forward to this, there's still no cure and no treatments for mitochondrial diseases, and this is a prevention, but there's still no cure and no treatment, and the Lilly Foundation are not going anywhere until we make progress. And Sarah and Sandy have asked me to talk briefly about our plans for the next 10 years. And our 10-year vision is to create a UK Mitochondrial Disease Research Institute. This will be driven by Lilly. It will be guided by the experts. It will be shaped by the patients and families, and working together, we will hopefully find cures and treatments for this disease. We hope, but by 2035, we won't be talking about mitochondrial disease as incurable. We'll be telling the story about how we cured it. So in the UK, the situation at the moment is that we do have world-class scientists and passionate clinicians. We have powerful tools like AI. We have a very and deeply engaged patient community. We have the biosources, animal models, and patient data. The UK is making strides, but we are being held back by one simple truth. And that is that they are not yet working as one. And only by bringing these together will we finally unlock their potential and that cure. What is really needed now is long-term sustainable investment in order to build the infrastructure and the systems that will lead to breakthroughs. So taking a closer look, the Institute will create a fully operational nationwide patient registry, a comprehensive database that will tell us who has mito, what syndrome they have, where in the country they live. It will link clinical data to genetic information and treatment outcomes. It will also integrate with biosamples, allowing for greater understanding of how the disease behaves and where the most urgent needs are. This will give researchers clarity, and pharmaceutical companies are ready to go to landscape, giving patients confidence that their data is actually driving genuine discoveries. And when it comes to patient samples, we have many across the country. We have blood, tissues, biopsies, but they're often scattered, unlinked, and worst of all, sometimes wasted. So the Institute will create a centralised, accessible biosource matched with patient data, ensuring samples are put to work where they matter most, accelerating drug development, biomarker discovery, and personalised treatments. No more missed opportunities, no more untapped potential, just fuel for breakthroughs ahead. Across the UK and beyond, we know there are powerful animal models, from mouse networks to zebrafish models, that mimic human disease and are key to testing new therapies. The Institute will make this a nationally discoverable, shareable and integrated so that researchers can collaborate, compare and accelerate testing, bringing treatments closer, faster. We've all seen how AI can transform our everyday lives. It's already turbocharged diagnosis and drug development in other areas of disease. And in mito, it's barely begun. With proper infrastructure and data, we can harness the power of AI to diagnose faster, identify drug treatments more effectively, and even predict treatment responses. AI won't replace scientists. It will just supercharge them, helping them to do what they do more quickly and more effectively. And finally, I've already given it away, none of this can happen without our people driving it all. The patient community of scientists, clinicians and patients are our greatest strength. For the current generation of mito researchers, sorry guys, but not getting any younger, and for our next generation, the path ahead is uncertain. A lack of long-term opportunities means that we are risking losing but really promising early career researchers to other countries or better funded sectors. The Institute will be a powerhouse for talent, a place to mentor, to train and to inspire, where research will not just survive, but it will thrive, where brilliant minds will be equipped with the right tools, where early career researchers are supported and the senior leaders stay involved. We're not just recruiting homegrown talent, but attracting it from across the globe, where collaboration, not competition, will define our culture. This isn't just about retaining talent. It's about creating the future generation of leaders who will drive mitochondrial science into the next decade. So individually, these elements are powerful. Together, they're transformative. And what we're proposing isn't a collection of siloed projects, but an entire ecosystem that allows mitochondrial disease to grow and to thrive. A purpose-built resource that eliminates duplication and accelerates discovery, cements our place as global leaders in the field, and gives families affected by mito something they've never really had: a clear, coordinated path to treatment and to hope. Because it's only by really seeing that full picture that we would truly understand and be able to treat mitochondrial disease.
Joining us from Melbourne, Professor Catherine Mills. Catherine is Professor of Bioethics and Reproduction in Society Research Lead at Monash University. She is Patient and Community Engagement Lead at the mitoHOPE pilot programme for mitochondrial donation over in Australia. And she's the author of books including Biopolitics, Futures of Reproduction, and a contributor to books including Reproduction Reborn.
So look, it's really great to be here and great to have a chance to talk through some of the ethical issues. I know everyone will be aware that there have been long-standing ethical discussions around this technology. There's been quite a lot of controversy at times. But rather than kind of rehash some of those debates that are already in the literature or some of those concerns, I want to shift focus a little bit from thinking about the kind of theoretical ethics and theoretical discussions that have happened so far to thinking more about ethics in context and thinking about what do we actually need to be dealing with as we move through the process of implementation of technologies like mitochondrial donation. So I'm really going to broaden the perspectives here from the kind of technical aspects and research and clinical aspects that Mary and Doug spoke about to really think about whether this technology works ethically in society today and how do we get it to work ethically if it's not quite there yet. So this is really, to my mind, a question of legitimacy. Is the technology gaining legitimacy throughout the implementation process, or what more do we need to do to be able to attain that level? And I think trying to kind of think about the kind of legitimacy of a technology like mitochondrial donation is very complex because we're always dealing with kind of complex trade-offs and getting the balance of trade-offs around various interests and stakeholder views and so on right, which I think is really essential to achieving legitimacy in the long term. And I think legitimacy is where is important for everyone. I mean, it's important for people within the mitochondrial disease community. People who use this technology don't want their children to be stigmatized. It's important for the scientists and the clinicians involved in the technology. It's important for bioethicists such as myself. So it's broadly important as a kind of social phenomenon to get innovative technologies to a position of legitimacy. So I think for tonight, this is a complex phenomenon, I will say, but I'm just going to very briefly touch on three particular areas of trade-off that I think have been important for mitochondrial donation so far and will continue to be important. So the first of those is the question of the pace of innovation. The second is around justice and resource allocation, and the third is in terms of equity of access. So these are not the kind of typical ethical issues that get taken up in the literature, as you might be aware. So just first, in terms of the pace of innovation, I started thinking about this because we get kind of two different responses in the media, I guess, to the announcement in July around the children born through the Newcastle program. So on the one hand, there's a question, or some people make the point that actually the program has been very slow and we've only got 8 children and this is actually not very much to show. Other people worry that actually it's moving at a much too fast pace and actually there needs to be a slowing down because we haven't yet established safety. I think there's a kind of interesting ethical tension there between how we think about speed and how we think about safety and how we bring those two things together. One thing that's worth remembering on the question of moving too quickly is that, in fact, research in this area has been going on for decades. So preclinical and scientific research has been going on for decades, and that gives us a very good, solid foundation for understanding some of the things that will happen when using this technology. Yes, they're in animal models, but some of that science, at least, is transferable to humans. And now we're in the very beginning stages of actually trying to use this technology in humans, hence the need for appropriate safety and strong kind of criteria around access and legitimate eligibility for using this technology. Hence, not many people are using it at this point, a point that I'll come back to later. So one of the other things that is often mentioned in relation to this technology is the question of the monitoring of children and people born from the technology afterward. How will we know that the technology is actually safe unless we undertake long-term monitoring? So the worry there, of course, is that we can't really know whether this has worked even just within that 5-year period of monitoring, because who knows what will happen afterward. That's not an illegitimate concern, but I think it also has to be balanced with the concern that people in the mitochondrial disease community have around excessive medicalisation, what Liz mentioned as being treated as an experiment. Of course, no one wants that, but I think precisely what we need to be doing here is actually developing models for ongoing monitoring that avoid that medicalisation. And I think the kind of national registry model that Liz actually elaborated would be the perfect way of doing that. So you're not kind of continuously testing, but actually using the technologies and capacities that we have these days to actually build long-term registries that don't impact on people's lives too much. Now, obviously, I'm just speculating here about how we could do that, but I think it is something important that we need to think about of how we do monitoring. So really, the core question when it comes to pace of innovation is really just how much uncertainty are we willing to accept when reproductive lives are at stake? And I think it's really important to recognise that that will be different for different people depending on the stakes that they have in how that decision is made. And I think we always have to keep that in mind when we're talking about people's reproductive lives, because people will value different things differently. So let me go on to justice in resource allocation, because this has been an emerging issue in the kind of ethics literature around mitochondrial donation. And the question is simply, so we have limited healthcare resources. We all know that. We know that the NHS is underfunded. We know that health is underfunded in Australia. So is a technology like mitochondrial donation, which may affect or may benefit quite a limited number of people, is this really the way to spend our precious health resources? And there's two kind of issues here that I think we can immediately go to that or are sometimes raised in relation to this technology. The first one is the distinction between treatment and prevention. So many people point out that this is not, in a sense, a treatment. It's certainly not a cure. It's not a treatment because it's not treating any particular individual. But I think we have to, and that's fair, that's fine, that's a kind of interesting and important philosophical point, but it doesn't necessarily in itself undermine the justification for spending in this area. This is a complex condition, often have very severe effects on people's lives. Many rare diseases actually fall into the same bucket of costing significant amounts of money to treat, but are only benefiting a few people. And yet it would be completely unjust to provide no medical treatment for rare diseases. Now, we know that at this point, mitochondrial donation can't be seen as a form of preventative technology, but it is aiming to be a preventative technology if we can get the technique working well enough. So at the moment, it's a risk reduction, but from the evidence that we currently have, it works pretty well as a risk reduction technique. So then another way of thinking about justice in resource allocation is in terms of costs and benefits. And people will point out that so far, you know, the UK program cost about 8 million, I think is the figure that's often cited to set up. And what, we've got 8 babies. But that's a very reductive and simplistic way of thinking about the cost and the benefit of this program, because the benefit of what it means for the people whose babies have been born with significantly reduced risks of having mitochondrial disease, it's very difficult to capture how much benefit it is to them. It's also difficult to capture the benefit it will be to those 3 or 4 girls who were born who will themselves be able to have children without the risk of passing on mitochondrial disease to their children. So there are generational benefits that are rarely taken into account when we think about this. I think the final thing I will say here too is that we can't imagine that we will finish where we start, by which I mean that because there are 8 children born now, we can't imagine that there won't be many more than that born in the future when this technology is more efficient, when it's shown to be safe, and it's shown to be beneficial for people. So you can imagine that more people will be accessing this technology in the future. So it's at the moment very difficult to actually kind of do a cost-benefit analysis, I think. So one question that has been raised is the question of should it actually be publicly funded? So at the moment it is in the UK through the NHS, it is in Australia as well through the clinical trial program, but it's not, of course, clinically available in Australia at the moment. Nevertheless, my team and I undertook some social research on just this question, asking average people around Australia in what are called citizens' juries how they think about the question of public funding. As it happened, people who had never previously heard of mitochondrial donation, after 2 days of having it explained and thinking about the complexities of this, almost all of those people agreed that it should in fact be publicly funded. And so I've just got the reference to that paper, which will soon be coming out in BMC Medical Ethics, hopefully soon in any case. So my final point arises around equity of access. There are always concerns about disparities and potential exclusions when it comes to innovative technologies or health technologies in particular. So at the moment, we only have one program in the UK and an emerging program, a clinical research program in Australia. So there has to be limits on both who can, or there are, by virtue of there just being these single programs, going to be limits on who can actually access the program. But it's also important to note that this question of equity of access relates back to safety. So at the moment, we have quite strict eligibility criteria for accessing this technology, both in the UK and in Australia. We have an arduous and rigorous process for actually being able to get to use mitochondrial donation in both programs. But as we get more data on the safety and the efficiency and efficacy of this technology, it may be that we can actually broaden those eligibility criteria. And then there will have to also be a kind of consideration of the process for determining eligibility. So it seems to me that what we want to say is that ethical implementation means fair access for all who need it. And at the moment, there are very good prudential reasons for justifying the current limitations on who can access the technology, but we have to acknowledge that this may in fact change when we have more data on safety. So just to conclude, I think what we're actually striving for here is a bridging between science and society to try to establish or to think about how technologies achieve legitimacy. And to my mind, ethical implementation is key to establishing legitimacy, and it makes innovation meaningful. I would say that because, that's what I do. But it seems to me that MD's success or mitochondrial donation success depends not just on the science. It's not just about how well the technique works, how good the science is, and so on, but also on how that technology is actually introduced in the social context in which it's made available, how the regulatory processes happen, and how everyone with certain, or as many people as possible with pertinent views on the technology are actually involved in that introduction and implementation process. So I think achieving legitimacy is actually an ongoing collaborative process that needs to be achieved using appropriate balances across those 3 areas that I've talked about. And it involves not just the scientists, but also the patients, the advocacy groups, ethicists, regulators. So it's a broad cohort of people that need to be involved in this kind of ethical implementation of technologies like mitochondrial donation.
This is for you, Mary. What what does PNT stand for and what does PGT stand for?
Pronuclear transfer, PNT stands for pronuclear transfer, and then PGT stands for pre-implantation genetic testing. I'm sorry, I thought I had explained these in the talk. I'm sorry about that.
Not to worry, there's an awful lot to take in your talk, so you may have done, and it's just not been able to take it all in. And I suppose there's an anonymous question as well, is that sometimes it's called mitochondrial donation, sometimes it's called mitochondrial replacement therapy. Are we talking about the same things? Is one term better than the other?
In general, it's mitochondrial donation in the UK, mitochondrial replacement in the US. But I wouldn't call it mitochondrial replacement therapy because it's not a therapy. It's a treatment designed to reduce the risk. Yeah.
Does anyone else want to come in on that? Does anyone else have any preference on terms or think that one's better than the other? No? Great. Okay, we have a question from Lindsay Craven, who asks, can you tell us more about the UK regulator, the HFEA approval process? Will it be a similar process in Australia? And is there a reason for the drop-off between the number of HFEA approvals, 32, and ovarian stimulations, which is 25? So I don't know who wants to go first on that.
Right. So I'll do the approval process, and then I'll leave the difficult ones to Mary. What I'll do is so the approval process is very rigorous. Again, I'm sorry if I didn't explain this. So a specific form has to go to the HFEA for all patients who want to proceed with mitochondrial donation. This form has details about their variant. It has details of the clinical history. It has details of the family history, all of which are super important in mitochondrial donation. That then goes to a specific committee at the HFEA, and sometimes questions come back, which then have to be answered. And there is an external assessor. Before it goes to the committee, it's seen at the committee, and there's an internal assessor. I really feel that process, which has been put in place by the HFEA, is extremely rigorous and excellent. And it gives us reassurance that the patients going forward are appropriate patients. In the US and Australia, I'm not quite sure how the process is going to be done in Australia.
Before we go to Mary and perhaps Catherine to answer that bit, I'm just going to, there's a question from Russell Donis for you, Doug, saying, can you give us an overview of the criteria that must be met to gain referral for mitochondrial donation?
Well,
does the mother's blood and urine always get mitochondrial sequencing performed to confirm the mitochondrial mutation and heteroplasmy?
Absolutely. One, it's got to be a pathogenic mitochondrial DNA mutation because there's lots of variants in the mitochondrial genome. It's got to be at a level, and in the UK, you can only have mitochondrial donation if PGT is felt to be inappropriate. So that is a big criteria. And I think 3 patients now have gone from PGT to MD because the PGT didn't work for them. We definitely have confirmation of the genetic diagnosis in a diagnostic lab. This is a definite thing, and there's obviously got to be an appropriate family history. So the criteria are strict criteria that go forward. Most importantly, they've got to have a pathogenic mutation, and PGT, pre-implantation genetic diagnosis, is not appropriate for those patients.
Okay, so to go now to Australia and the approval process there. Shall I go to Catherine first for that, and then Mary, let you come in on that, and also deal with the drop in numbers from the approvals to the ovarian stimulations. So Catherine first.
Thanks, Sarah. Yeah, look, it's an interesting question. Australia doesn't have an organisation like the HFEA, so we have to have a different process, a slightly different way of dealing with it. What the program in Australia will have is an independent clinical advisory committee who will consider applications and make a recommendation as to whether that person should be allowed to progress to PNT or to mitochondrial donation or not. So that will be a completely independent committee separate from the program itself, who will consider the clinical aspects.
Great, thanks for that. And Mary, then, why is there this difference in numbers?
So 7 people who've had approval haven't yet come forward, haven't yet had egg collections. I can't say exactly why. I think there's a variety of reasons. Maybe some are still due to have their egg collections, or some went off waiting for a donor for too long, or so on. I can't give detail on that. I'm not aware. Sorry.
Thank you very much. Okay, the next couple of questions, I'm going to go from Dagan Wells. Dagan says the procedure succeeded in keeping heteroplasmy levels low. I'm not going to read all the numbers, it's quite a long question. However, 3 of the children ultimately had higher levels than this in their urine, and this appears to indicate a degree of mitochondrial DNA reversal. Do you think that this gives any cause for concern? I guess it emphasizes that given the limited data, mitochondrial donation should currently only be considered a tool for risk reduction and not a guarantee of a disease avoidance. Would you agree? So Mary first on this one, I think.
So definitely to the idea that mitochondrial donation is a strategy for risk reduction. And we've said that since we published our paper in 2016, when we first discovered that some ES cells can revert to the maternal mitochondrial genome. However, I think what we're seeing in these babies can be explained by carryover alone. So it's 12 and 16%. Now, if you look at the embryos, the spare embryos, the heteroplasmy in those varies from, I think it's around 3% in general, but that's heteroplasmy across the whole embryo. And we do know that there can be some unequal distribution of the maternal mitochondria within the pronuclear transfer embryo. So in that case, we can get some compartments with slightly lower levels. So if 3% across the board, you could end up with a 12% in some cell types if there's unequal segregation. So I don't think, I know we need to develop our vocabulary around this and indeed to understand the processes a bit better. But I think carryover alone would be sufficient to explain the 12 and 16%. But of course, we can't rule out at a certain degree. But I think Doug can speak to what are the long-term prospects for this 12 and 16%.
Yes, Doug.
I just wanted to reiterate that this, to the patients, this is always talked about a risk reduction. One thing you know in medicine is there's no certainty in anything that we do, so it's always going to be a risk reduction, and that's what's said. I also want to comment that as I've said, levels like this are a risk reduction, but if the levels stay the same, which we believe they will, and then the risk of actually developing disease is minimal to non-existent for the patient so far. There is, of course, the risk, and that if it's a female patient, the risks of possibly transmitting those variants to their children. So it's certainly an ongoing process of continued follow-up, which I think will benefit hugely when the Australian process gets going, because the numbers will help us here.
And so I think that sort of also answers a question from Bob Phelps about what are the lifelong prospects with the heteroplasmy of up to 16%? What are the lifelong prospects of contracting mitochondrial disease? And I think the bit, if one of these children were to develop mitochondrial disease, what support would they be offered, Doug?
Okay, right. So I don't, mitochondrial disease tends to affect tissues which are slowly dividing. I don't think there's any evidence of increasing levels of pathogenic mutations. in the patients we've seen over years, and therefore the same with these. So I think the risks are minimal. We will offer to follow up these children for as long as they want. Of course, it doesn't mean that they'll take up our offer. The second thing is in the UK, and we're very fortunate, I just keep stressing this, and I know Catherine mentioned about the NHS. In the UK, we're able to offer a national service for patients throughout the UK for patients with mitochondrial disease. So they will be followed up in the clinical service. So they would get exactly the same care as any other patients if under the remote possibility they develop disease.
Great. Thank you very much for that. Okay, I'm going to move on to Dagon's other question, because it's the most popular one, which is, it's worth noting that 8 births required more than 500 donor oocytes rather to achieve. Given the cost and or restricted availability of donor oocytes, a requirement of more than 60 donor oocytes per pregnancy will likely place limitations on the availability of the treatment. Could further technical efficiencies in the procedure be found? What do you think, Mary?
That's a strange way of looking at it, because we didn't require 60 eggs per pregnancy in the nature thing. So we had transfer procedures altogether and 25 donors. And the nature thing is, so we had surplus donor eggs, and they were used for training or for ongoing research subject to consent from the donors. So they weren't wasted eggs. So in terms of the requirement for 60 eggs, it's not quite correct to say that. But having said that, there is scope for efficiencies in this, and I think we could have efficiencies if we can use frozen donor eggs. And we're hoping, there's a reason we don't do that, is because it gives unpredictable carryover. So we're hoping our research will move us in a direction where we can use frozen donor eggs. In addition to that, when we're thawing patient eggs, we just do usually 10 at a time. And so that if we can do more eggs in any one go, then I think that will increase the efficiency in the use of donor eggs. So I hope that answers the question, but I would caution against saying we require 60 eggs per baby. The surplus eggs were used for other purposes.
Thanks, Mary. And I suppose a question Sorry, Mary, go on.
I also wanted to say that the donors can be recruited by the patients or can be recruited by the clinic, and they're all counseled about what they are.
Excellent. That's preempted the next question I was going to ask you from Alison Lashwood. So yes, Alison, they are informed of their use. They're properly consented, which is a relief. And I assume, Catherine, that would be a similar process in Australia.
Absolutely. So we have to have quite a rigorous consent procedure in Australia to allow women to donate to mitochondrial donation. And it's very explicit consent for the procedure itself, as well as for donation.
Great, thank you. I think, when we haven't had one for a while, but when it was going through Parliament, we did actually get calls in the office from women wanting to donate their eggs, which was very nice. We referred them to Newcastle. So yes. The next question is from Dimitri Kalias. Apologise if I've mispronounced your name. Again, it's for you, Mary, in the first instance, but others might want to come in. PNT and MST, so pro-nuclear transfer and maternal spindle transfer, were developed to reduce the risk of transmitting mitochondrial disease, but are also being offered as infertility treatments in the IVF industry in some countries. What's your opinion on this?
Okay, hello, Dimitri. So my opinion on that is that based on work that Dagon Wells published, that actually it appears to be successful, but I think we need caution here, because we need to define under what conditions it's useful. And people immediately begin to think, oh, it might alleviate the risks of the maternal age problem with maternal age. And that's actually unlikely to be the case, because by the time they get to the pronucleate stage, most of the problems with maternal age are linked to chromosome segregation errors, which are already present at the pronuclear stage, and in many at the metaphase 2 of uterine spindle transfer. So I really would cover age-related infertility, and if anybody's making the claim, they should do the research first.
Thank you, Mary. Sorry about that. I lost sound for a moment, so I thought you'd finished and then was panicking so I couldn't speak. Okay, so a question from Ghulam Bahadur. I'll go to Catherine first with this one. Should the birth certificate name a third parent?
Interesting question. Well, actually, I mean, it's a very interesting question, actually, because parenthood is quite an interesting concept. But I think both the UK and Australia have determined in law that the donor is not a parent. And that's consistent in Australia with how it treats all gamete donors. No one who donates an egg or an embryo is considered to be the parent of the child. Similarly, in the UK, the gamete donor is not considered to be a parent. So the way in which mitochondrial donors are actually treated is consistent as that. They are not legally parents by any means. Some people have argued that they are considered or can be considered to be genetic parents, but very, very few people, when we've done surveys on this, very few people who donate eggs see themselves as having any parental role. I did notice that one question actually is about donor identification, and that's an interesting difference between the UK and Australia. So in the UK, there is no donor identification for people who donate mitochondria. In Australia, that is different. So in Australia, we will actually have a system whereby children or people born through mitochondrial donation will be able to find out identifying information about their donor. But they're not parents and have no capacity to become parents, legally speaking.
Great. Okay. Does anyone else want to come in on that? Or are we happy to have left that to Catherine? Looks like happy to have left that to Catherine. Okay. So the next question I'm going to go to is one for Mary, but it's probably one for all of us, really. And that is from Bob Phelps. And he says, in the media, Mary agreed mito replacement only reduced the risk of mito transmission to children, not prevention. 100 Stories globally misled the public, saying mito was prevented in UK children, and this was repeated. Now, how will PET, and the researchers correct the record and explain their lifelong risks? So, Mary, if you want to go on this one first.
I just put this reducing risk in perspective. I think the reducing risk refers to the fact that we think it may not work in all cases. There may be cases where we don't manage to reduce the risk. However, in these babies that were born, it's highly unlikely that they will develop mitochondrial DNA disease. And I think, therefore, I think Doug would agree with this. We can say that in those cases, it would not be untrue to say that it has been prevented. Doug might have more to say to that.
Yeah, I mean, I'd agree. I think there is, as anybody that's been involved in this, the way in which things are reported around this particular story knows that you can have things get into the story. It's very difficult to change the narrative sometimes. Yeah, I mean, there are lots of people walking around with levels of pathogenic mitochondrial DNA mutations who do not get disease, and in much higher levels than these patients, the children. I think we prevented disease in these children. I think we would be we can't be 100% sure because we've never followed these children up to the end of their lives, but we are. The evidence that we have, and all the evidence so far, is that we prevented disease in these children.
Catherine, did you want to come in on the importance of language? It was of interest to bioethicists.
Yeah. Look, I think it's I mean, certainly in my experience, everyone that I speak to involved in the development of this technology is very careful to maintain the language of risk reduction. And certainly within the program in Australia, that has been a very consistent line that we have taken. We have not been promoting it as a kind of preventative technology. I think ultimately we would like to get to a point where we could consider this a preventative technology, but we are all aware that at the moment there's still work to be done to achieve that. So I think there's very little you can do to correct the record in the media these days. That's just the nature of global media. But the people who are working in this space have been very careful to use the language of risk reduction.
And Liz, how's it talked about at the Lilly Foundation among the people there that you support?
The families were delighted with the news, and I think, as I said earlier, we've seen a big shift in people talking about it, thinking about it as a potential option, whereby previously, without the knowledge that it's worked and that the babies are healthy, it's a much more difficult decision to make. So yeah, I'm hopeful that it will become an easier process in the long term for patients to be able to take up.
And speaking on behalf of PET and the use of language, we've always been really careful to say avoid transmission. Actually, it's something that I've, even with my misspeaks and getting dates wrong, I am very careful to do. Okay, the next question is one from Bernard Fox. I don't know how you pronounce with the umlaut over the E. Apologies. If mitochondrial DNA comes from the mother, why are apparently healthy mothers having children with mitochondrial disease? Surely they should carry the same mitochondrial DNA. So I think perhaps go to you, Doug, first.
Yeah, it's like any variant. You can get spontaneous mutations. There are sporadic mutations of mitochondrial DNA. Perhaps the most common of those are deletions of mitochondrial DNA, and they form a significant number of the patients that we see in our clinic. So it's like other genetic diseases. You have sporadic variants. Sometimes mothers can carry relatively low levels of mitochondrial DNA variant and be perfectly healthy themselves and have children that have higher levels. Those are precisely the sort of precisely the women that would do really well with PGT. So I think either it's a sporadic or it's at low level in the mother, below the level of symptoms, but because of a phenomena in the transmission of mitochondrial DNA, the bottleneck, which I'm certainly not going to try and explain over a video call, you can have higher levels in a child, and I think those are the two reasons.
Thank you. Well, maybe not a quick one for you, Mary, but are there any plans to explore other potential types of mitochondrial donation, such as polar body transfer?
So the two types that are currently approved are spindle transfer and pronuclear transfer. Polar body transfer has been done on a research basis. There are some papers out there with that. It certainly is an appealing idea in the sense that the polar body is naturally extruded by the egg. So in a sense, it's a ready-made carrier containing a maternal nuclear genome. But I think more work needs to be done. It looks to me like the spindle is very slow to assemble when you put this. This is if you like is the polar body is a non-viable product of female meiosis. So it contains a maternal genome, but it's not in the spindle. It's essentially discarded, and to resuscitate that and to get it to produce to develop to organize a normal spindle, the machinery required to divide the chromosomes properly. I think that will take further work, is my take on what's the research that's been done there. But in any case, it's as far as I know, not up for being licensed in either the UK or in Australia for sure.
So, Catherine, is it the same in Australia that the techniques that the scientists can use here is limited to pronuclear transfer and maternal spindle transfer?
Yeah, so Australia has a complex licensing system in place that we're navigating at the moment. And each technique that we propose to use has to be licensed separately. And at the moment, it's only possible to attain licenses for PNT and MST.
But we are allowed to do research
for human use, yeah. Yeah. Sorry, Mary.
Transfer. We are allowed to do research on polar body transfer, and it is worthwhile pursuing.
Yeah, it is certainly in research, but in the clinical context, not at this point. Yeah.
Liz, I'm going to go to you first with this question, and then allow Doug and Mary to chip in. Did it upset you when people call this Frankenstein science in the media?
Yes.
Yes.
It was one of the first reports, right? It was on the Observer on a Sunday. It was really quite disturbing. And Science Media Centre then, I think, rescued the situation. I think it's fair to say that was my take.
Yeah. And how about you, Liz?
Yeah, as Mary said, I think it was one of the first reports that I was aware of, and I think it certainly laid the path to the job that we had to try to undo the damage in the months to come, when we were talking to MPs and trying to educate them into what this science actually was, and the reasons, that Doug first talked about, that this first came into play. So yeah, it wasn't helpful, put it that way.
Have you had similar headlines in Australia, Catherine?
We haven't had so much. We certainly haven't had the headline, as far as I know, around Frankenstein science. But there has been some discussion around three-parent babies and so on. But I mean, fortunately, in Australia, the debates and the media language had moved on somewhat by the time we started seeing a lot of media in Australia, and so most journalists have been quite careful to use things like three-person IVF rather than three-parent IVF, for instance, which is good.